Hello, everyone. Welcome to this event in the International Lecture Series on Chromosomal Instability. What you see on the screen is our whole program for this year, and I hope you enjoyed Christina's and Sam's talks in January and March. Today we have Peter Lee, and I introduce him in a second. And if you stay fans of this series, you will be able to listen to Renata in July, Stamatis in October, and Aleph in November. The series is organized jointly by me. I'm Florian Markowitz from Cambridge, and Isidro Cortes-Siriano from EMBL EBI, and Aurélie Ernst from DKFZ Heidelberg. We're very happy to have a talk today by Peter Lee from UT Southwestern. Peter started his lab there in 2019 after a postdoc with Don Cleveland at UC San Diego. And we are very happy to have him here today to tell us about his recent work on mechanisms of aneuploidy. Peter, over to you.
Okay. All right. Thank you, Florian. Well, first, I want to thank the organizers, Florian, Isidro, and Aurélie for organizing this wonderful lecture series. It's an absolute pleasure to be part of this and share our work with you today about and tell you a little bit about what our lab is doing. So my lab is fundamentally interested in the mechanisms of genomic instability, particularly in the consequences, in the mechanisms and consequences in the development of cancer. So on the left is a spectral karyotype of a diploid human genome. And what you can see are 23 pairs of chromosomes whose copy number and sequence order has been evolutionary shaped or shaped over evolutionary timescales. Cancer cells, on the other hand, which is an example of which is depicted on the right, typically deviate from this norm and harbor a number of abnormalities, including alterations in both chromosome number in the form of aneuploidy, as well as structural rearrangements that can rearrange the linear sequence order of individual chromosomes. Depicted in these arrows are examples of translocations, for example, where you have rearrangements between two different non-homologous chromosomes. And another way, and one of the fundamental questions that our lab has been interested in is understanding how we go from a normal diploid genome into one that looks something like this on the right, and how this drives cancer development. So another way to look at these types of rearrangements are via circles plots. And so on the outer ring here are individual chromosomes, and lines that connect two different chromosomes represents an interchromosomal rearrangement. And additionally, you can also have intrachromosomal rearrangements depicted here by these lines that connect the same chromosome. And if we look at a cohort of cancer genomes, these are 9 colorectal cancer patients' genomes, what we can see is that there's abundant heterogeneity in different genomic patterns of copy number changes and also structural rearrangements. And what I really wanted to draw your attention to are these intrachromosomal rearrangements. So and that's depicted here in these red lines. And typically, when these form, they form in a very clustered and complex fashion. And these were discovered a number of years ago by Peter Campbell and colleagues, which we'll get into. And they termed these types of complex intrachromosomal rearrangements as chromothripsis, which is a Greek term for chromosome shattering. And that will become apparent why they call it that in a couple of minutes. So if we look at the signatures of these complex and localized genomic rearrangements across pan-cancer genomes, this is a very nice study from Isidro Cortes-Siriano looking at the pan-cancer analysis of whole genomes consortium datasets, looking at over 3,000 tumors. What we can see is that there are lots of chromothripsis events in different types of cancers. So for example, on the left-hand side, we have liposarcomas, osteosarcomas, and glioblastomas that has a particularly high prevalence of chromothripsis. There's also a number of other cancers that exhibit features of chromothripsis as well at varying frequencies. But across the board, it seems that the signatures of chromothripsis can be observed in approximately 30 to 40% of all human cancers. And so when chromothripsis was initially discovered by whole genome sequencing, it was presumed or predicted that these events arises from a chromosome shattering event in which you begin with an intact chromosome. This chromosome experiences something that causes it to shatter into multiple pieces. Most of these fragments actually do not have a centromere, and that will be important for the later part of the talk. And then the cells utilize their intrinsic DNA repair machinery to stitch these chromosome fragments back together to form this derivative chromosome that we often see in human cancers. And some fragments also fail to participate in this reassembly event and therefore become lost to the cell. And particularly, these could potentially hold or harbor tumor suppressor genes. In addition, we can also have the fragments that become circularized from the ligation of the two ends to one another or between multiple fragments, and they form these extrachromosomal DNA entities that were formerly referred to as double minute chromosomes that can potentially harbor oncogenes or genes that can confer resistance to drugs, for example. And when Chromothripsis was initially discovered, it was quite puzzling and also surprising, and it really challenged the textbook model of tumorigenesis, where it was canonically thought to arise almost exclusively from a stepwise pattern of gradual accumulation of different alterations over a long period of time. So for example, here you can start with a cell with a normal genome, and it accumulates and over time it accumulates different alterations to eventually result in these complex karyotypes that cancers usually have. But in the context of Chromothripsis, you can start with a normal genome or a normal chromosome, and it undergoes this catastrophic punctuated event in which it could rapidly reshape or alter individual chromosomes, and you can get the similar types of patterns at the end. And so one way to look at features of Chromothripsis from whole genome sequencing data is to plot each individual chromosome by its position from one telomere to the other telomere, and then to plot it by copy number. And so for a diploid chromosome, this is what it would look like. And after a chromosome has undergone Chromothripsis and has become rearranged, one would see signatures that look like this, where you can have lines that connect different pieces of that chromosome. You can also have chromosome fragments that are lost, which are depicted as a copy number decrease. And you can also have amplification of different segments in the form of ecDNAs, which are then depicted as at a high copy number. And this is a very simplistic overview or view of how of what Chromothripsis would look like. But if we look at an actual example from the initial paper from Peter Campbell's group, we can see that is extremely complex. So here's an example where there are multiple lines, potentially hundreds of lines that connect different segments of the same chromosome, and each line here represents a single structural variant. And of note, another hallmark characteristic are these oscillations in DNA copy number steps. So the DNA copy numbers oscillate back and forth between two steps. The upper copy number state represents regions of the chromosome that were retained in the chromothriptic chromosome, and the bottom segments represents regions of the chromosome that were ultimately deleted from that chromosome. And if we zoom out and take a whole genome view of this particular example, we can see that Chromothripsis of chromosome 5 is the sole cytogenetic abnormality that's present within this example from a kidney cancer tumor. And this really raised a number of questions. For example, how can all of these rearrangements be restricted to an individual chromosome, whereas the rest of the genome is essentially spared? And this presumably arises from some type of DNA damage. So shortly after the discovery of Chromothripsis, it was then hypothesized that chromosome segregation defects during mitosis could potentially drive or initiate these events that resemble Chromothripsis. So for example, here's a time-lapse movie of a cell in mitosis. You can see the chromosomes are aligned at the metaphase plate, and you have a single chromosome that fails to properly align itself. And when these cells enter anaphase and begin to segregate their chromosomes, this missegregated chromosome becomes encapsulated into this abnormal nuclear structure called a micronucleus. And this is a really attractive hypothesis for how you can get Chromothripsis, because now you can spatially isolate a single chromosome away from the primary genome, where it can presumably then acquire DNA damage. Indeed, in 2015, David Pellman's group used live cell imaging coupled with single-cell DNA sequencing to demonstrate that mother cells with the micronucleus, when they divide and give rise to two different daughter cells, one or both of these daughter cells can in fact acquire the signatures of Chromothripsis that very much resembles what we typically see in human cancer genomes. Although at the time, the mechanisms for exactly how chromosomes and micronuclei become something like this remain poorly understood. So one way that our lab has been interested in understanding and studying Chromothripsis and the formation of micronuclei is to induce chromosome-specific segregation defects. And in this way, if we know what chromosome is missegregated and inside the micronucleus, we can then track it over time, over multiple cell cycles and even further. And so I'll just very briefly introduce this system. And so here we're taking human DLD1 cells, which is a colorectal cancer cell line, p53 deficient, but also diploid. And we can engineer them to harbor a degron on a centromere histone H3 variant known as SEMP-A. And so SEMP-A typically localizes to centromeres and forms the kinetochores that can then allow chromosomes to attach to the mitotic spindle during mitosis. For reasons that are still somewhat unclear, the human Y chromosome is a little bit different from all of the other chromosomes. So the Y chromosome almost exclusively depends on one of the terminal tails of SEMP-A, whereas the rest of the chromosomes has this redundant mechanism for chromosome segregation by SEMP-A. And so here, what we did was we took the cells and we degron tagged the endogenous SEMP-A, and then we introduced a mutant version of SEMP-A that fails to support assembly of the kinetochore, specifically on the Y chromosome centromere. So we can take these cells, add doxycycline and auxin to them, and replace SEMP-A with this mutant version, and essentially turn the Y chromosome centromere from an on state to an off state. And this induces very high frequency of Y chromosome segregation errors that then results in their entrapment in these micronuclei. So here's an example of this. So this is a cell and interphase in which we can use DNA fluorescent in situ hybridization probes to mark the Y chromosome inside of this micronucleus. These Y chromosome containing micronuclei acquire all the different signatures of micronuclei that many other labs, including ours, have demonstrated, including the acquisition of a large number of DNA double-strand breaks indicated here by staining for gamma H2AX. One question that we wanted to ask first is what happens to these chromosomes when these cells enter the next mitosis. And to do that, we can simply collect metaphase chromosome spreads and use FISH to look at what happens to the Y chromosome. So here's an example of what happens. So this is a metaphase spread from a cell after induction of Y chromosome micronuclei. And you can see in blue are your normal nuclear chromosomes stained with DAPI. And then in green are fragments of the Y chromosome after the chromosome had shattered into multiple pieces. And this has now become a really powerful system for our lab because we can induce in a very controlled way the induction of Y chromosome micronuclei and then look at what happens to it over the next cell cycles, including measurements of how these chromosomes undergo fragmentation. And this assay, we can both quantify how frequently this occurs and also the severity, but just by counting up the number of different fragments. Okay, so just to kind of recap what we know about micronuclei, they typically form from errors during mitosis in which you can get lagging chromosomes or misaligned chromosomes, or also acentric chromosomes after DNA damage. And these cells, when cells undergo anaphase and segregate their genomes, they can form into these abnormal nuclear structures called micronuclei. And over the next interphase, these micronuclei acquire all sorts of very interesting characteristics. So this includes rupture of the nuclear envelope. So the nuclear envelope fails to properly reassemble around these missegregated chromosomes, and they can be deficient in specific nuclear pores or specific lamins. And this predisposes them to this process of nuclear envelope rupture, which is permanent, at least for that particular cell cycle. After nuclear envelope rupture, you also have a loss of nucleocytoplasmic compartmentalization, such that nuclear contents that initially were present inside this micronucleus spills out into the cytoplasm, and conversely, components that are in the cytoplasm can now freely enter this micronucleus and potentially interact with the underlying DNA. There's also a very strong DNA replication defect. So for example, if you feed cells a nucleotide analog and look at how chromosomes and micronuclei replicate versus the primary nucleus, they can either replicate very slowly, late, or asynchronously from the primary nucleus, or not replicate at all. So there's this very robust replication defect that's present in interphase. And additionally, the underlying DNA can also be subjected to nucleolytic cleavage by different types of nucleases that are present within the cytoplasm. One potential candidate is known as TREX1, which can potentially induce DNA double-strand breaks also on chromosomes and micronuclei. And additionally, once the nuclear envelope ruptures, this can terminate transcription and give rise to the accumulation of RNA DNA intermediates or RNA DNA hybrids that can become processed by the base excision repair pathway to generate both single-strand breaks and also double-stranded breaks. And so collectively, how all of these work together to induce chromothripsis has remained somewhat unclear. And so what Justin in my lab, who's a very talented graduate student, wanted to set out to do is to identify genetic pathways that can regulate chromothripsis from micronuclei. And we wanted to take a CRISPR-based screening approach. And for those of you who are doing CRISPR screens, know that one of the most essential components of a good CRISPR screen is having a really strong readout for your particular phenotype. And so one question is, how can we establish a CRISPR screen using the tools that we've built using the Y chromosome system? So we wanted to actually couple the induction of chromothripsis with cell viability and use this as a potential readout for a CRISPR screen. So previously, when I was a postdoc, we had taken this Y chromosome system and we engineered them using CRISPR editing to harbor a neomycin resistance marker on a specific location on the Y chromosome. So now we can induce Y chromosome missegregation into micronuclei, have the chromosomes fragment into many pieces, and now one or more of these fragments will then contain this neomycin resistance marker that can provide resistance under selection with G418. And so using this approach, we can look at different conditions using a clonogenic assay and show that under control conditions, for example, if you select with antibiotics only, or if you just simply inactivate the Y centromere, nothing really happens to cell viability. But if you inactivate the Y chromosome centromere, the vast majority of these cells, about 90% of them, actually lose the Y chromosome because they don't normally encode any essential genes. However, if we now combine these two conditions together, we can isolate a subset of cells that all contain the Y chromosome within these cells. And we've previously characterized these extensively by both cytogenetics and by whole genome sequencing to demonstrate the features of chromothripsis that are present within this fraction of resistant cells. So now we have a really good system where we can couple chromothripsis, the induction of chromothripsis, to cell viability. And so what Justin did is he took this platform and then he introduced a DNA damage response CRISPR screen that targets approximately 300 or so genes in the DNA damage response. And then he separated these into two different experimental arms. The first was a control arm in which micronuclei was not induced, and then the other arm was a condition in which we've induced Y chromosome micronuclei, and then we've added and then we've selected for these cells using G418, and then simply measured the abundance of guide RNAs at the final time point. And so from this screen, there was a number of surprising hits that emerged in both directions. So there are genes that become enriched. So these genes, if you delete these genes, the cells somehow survive better in the presence of G418. And then on the other side, there are depleted genes in which if you inactivate these genes, the cells survive worse in the presence of G418. And what really caught our attention was components of the Fanconi anemia pathway that all emerged on the side of enrichment. So and I'll get into this in the next slide, but there are multiple factors that came out from this specific pathway that came out from the screen where it appeared that if you delete any of these Fanconi genes, then you can inhibit some step of chromothripsis. But at this time, it was we had no idea exactly which step was inhibited. So the Fanconi anemia pathway typically resolves DNA interstrand crosslinks and replication and recombination intermediates. And so just to very quickly summarize this pathway in a very simplistic way, there's this Fanconi anemia core complex that's comprised of a large number of different proteins that functions as an E3 ubiquitin ligase to monoubiquitinate this heterodimer of Fanc-I and Fanc-D2. And so once Fanc-I and Fanc-D2 are activated by monoubiquitination, they can recognize different types of DNA lesions, potentially interstrand crosslinks or even different replication intermediates, and then bind to this DNA and then execute and then initiate the execution of a number of different pathways downstream that could then potentially repair that particular lesion. So we first wanted to ask, how does the Fanconi pathway, where exactly is it functioning in this pathway from micronuclei to chromothripsis? And so what Justin did was he deleted two different components of the Fanconi pathway, either FAP20 or Fanc-D2, and he first looked at nuclear envelope rupture using this fluorescent protein tagged to a nuclear localization signal. So what he saw is that in wild type and Fanconi deficient cells, the nuclear envelope of these micronuclei ruptured at the same frequency. In addition, if we looked at other characteristics of micronuclei, including the acquisition of DNA damage by staining for gamma H2AX, or looking at the replication defect by feeding the cells a nucleotide analog called EDU, we can see that DNA damage is still present within Fanconi deficient cells, and there's also this very strong replication defect. And so what we were able to very quickly rule out is that the Fanconi pathway does not contribute to an interface defect within micronuclei. And so next, Justin looked at mitotic chromosome spreads to look for chromosome shattering, and what he found was actually quite surprising. So here's again that chromosome fragmentation assay that I had showed you. This is an example of a normal Y chromosome using two different FISH probes targeting the Y. And this is an example of a fragmented Y chromosome in control cells. And then if we now look at Fanconi deficient cells, what we see is that the frequency of fragmentation is now reduced by approximately half. And in addition to this reduction by half, we can also see a reduction in the severity of the number of fragments that are generated per event, suggesting that the chromosomes shatter less frequently and does so less extensively as well. And so what we had then proposed is that there's potentially two different processes that could drive chromosome fragmentation. The first is a Fanconi dependent fragmentation that gives rise to this more extreme fragmentation that produces somewhere between the range of 10 to 100 fragments per event. And what I'll show you in the next couple of slides is we think that this can be pinpointed to this replication defect. In addition, there's also this Fanconi independent event that can give rise to more minimally fragmented chromosomes, and this presumably arises from DNA double-strand breaks that begin to accumulate within micronuclei during that previous interphase. And so one question that we wanted to ask was how exactly are these chromosomes becoming shattered after activating this Fanconi pathway during mitosis? And so after the Fanconi pathway is activated, it can recruit a number of different proteins to the lesions, one of which is this nuclease scaffold called SLX4. And this sort of functions as a Swiss Army knife that can recruit multiple nucleases to different lesions. And so, for example, SLX4 can recruit the XPF ERCC1 endonuclease, MUS81 EME1, as well as SLX1. And so we wanted to first see if any of these nucleases were responsible for inducing chromosome fragmentation. And so Justin first simply just depleted each of these components using RNAi and saw that depletion of XPF was the only nuclease that was sufficient to recapitulate the loss of the Fanconi pathway in inducing or in minimizing chromosome fragmentation. Justin next took cells and deleted XPF using CRISPR, and in multiple clones using multiple guide RNAs, we also see the same effect. And in addition, we also can delete SLX4 and also see the reduction in chromosome fragmentation. This reduction can also be rescued if we introduce a wild-type copy of SLX4, but not a rescue version of SLX4 that contains a mutation in its MLR domain. And this is the domain that recruits XPF ERCC1. So this suggests that the chromosomes, that the cells enter mitosis and that these cells undergo nucleolytic cleavage by the SLX4 XPF ERCC1 endonuclease complex. So what's so special about these chromosomes that makes them engage the Fanconi pathway during mitosis and undergo cleavage? So Justin did a series of very simple experiments that were very revealing. So first, he took cells and he simply fed them EDU, the nucleotide analog, for one complete cell cycle, and then he looked at mitotic cells. So if you look at control cells, you can see that EDU incorporates into all of the chromosomes. In intact Y chromosomes, marked here by FISH, EDU also perfectly incorporates into this chromosome as you would expect. However, now if you look at conditions where you have fragmented chromosomes, and this is in wild-type cells, then you can see that you can see the fragments illuminated here by FISH, but all of these fragmented pieces now fail to incorporate EDU, suggesting that the fragmented chromosomes typically arises from an underreplicated chromosome in the previous cell cycle from that micronucleus. And this is the quantification here, in which there's a reduction in this EDU intensity. And in addition, Justin next did another complementary experiment. Instead of feeding the cells EDU during one cell cycle, he fed the cells as they entered mitosis. And what he saw was that chromosomes that had undergone fragmentation now begins to replicate complete replication of their DNA, specifically during mitosis. So they undergo this mitotic DNA synthesis process. This is the quantification of this, in which we can see that the fragmented pieces incorporate much more EDU during mitosis, and this occurs in a Fanconi-dependent manner. So if you knock out FANCD2 or any other component of the Fanconi pathway, then you can reduce the ability of these cells to complete replicating that under-replicated chromosome during mitosis. So this is sort of a simplified model that we had drawn up. And so here you have chromosomes and micronuclei. they acquire different types of DNA replication defects during interphase. And when these cells enter mitosis, this activates the Fanconi pathway via the ubiquitination of Fanci and FANCD2. And then this induces nucleolytic cleavage of the chromosome, specifically during mitosis through the action of this SLX4 XPF ERCC1 endonuclease. And one prediction is that these events would then give rise to genomic instability in the form of complex genome rearrangements and potentially extrachromosomal DNA. And so to first look at genome rearrangements, we use this assay, the cytogenetics assay that we had developed in our lab, in which we can take the Y chromosome and label them with two different FISH probes. The top probe labels the first half of the Y chromosome, the second probe labels the second half, and this is what a normal Y chromosome would look like. And so after a simple rearrangement, such as a translocation, it would look something like this. And if you have a complex rearrangement, such as Chromothripsis, it would then look something like this, where the signals now completely merge or co-localize together. And so using this assay, we also demonstrated that if you inactivate both either FANCD2 or XPF, this is sufficient to suppress the formation of both simple rearrangements and complex rearrangements after the induction of micronuclei formation. And so we wanted to take this one step further and look at this in a more clinically relevant setting. And so we collaborated with Roger Lowe at UCLA. And so Roger's lab had previously demonstrated that melanomas that undergo targeted therapy with BRAF inhibitors and MEK inhibitors can become resistant to these inhibitors over time. And this is typically through a chromothripsis event that generates ecDNAs that confers resistance to these drugs. And so we wanted to test whether the Fanconi pathway is required for this. And so here we deleted Fanconi, the FANCD2 in a number of different melanoma samples. And what we can see is that under control conditions, there's really no changes in viability. But if you treat these cells with with a combination of these targeted therapies, that over time, most cells die, but then resistant cells begin to emerge over the period of about 1 month. And if we now delete the Fanconi genes, we can see that we can suppress the formation of the emergence of these drug-resistant cells. And if we now collect up all of these different cells and look at their karyotypes and stain them with the sensitive DNA stain called yo-yo-1, we can see that in wild-type cells that develop resistance, there are these extrachromosomal DNAs that are apparent by this staining here. But if you now delete the Fanconi pathway, you no longer have the emergence of these ecDNAs. Okay, so next, one of the other major questions that we were really interested in is after a chromosome shatters into multiple pieces during mitosis, what actually happens to this chromosome? So what we can see if we included a centromere FISH probe on these fragmented Y chromosomes, we can see that most of these DNA fragments actually lack a centromere, so therefore they're acentric and cannot attach to the mitotic spindle. And so one outstanding question that Yufeng Lin, a very talented senior scientist in my lab, set out to address is how do these acentric fragments from shattered chromosomes segregate during mitosis if they cannot attach to the spindle? And so to do this, she wanted to develop a system where we can watch chromothripsis events happen in real time, and to do this by live cell imaging. And so what she set out to do was take the Y chromosome system that I had just described to you, and she now used a nuclease deficient dead Cas9 fused to a SUNTAG. And so each of these SUNTAG entities can recruit up to 10 or so copies of GFP. And so using this approach, we can now target this dCas9 SUNTAG to this repetitive sequence on the Y chromosome. And this is about 30 megabases or so on the Q arm of the Y, and remarkably using just a single guide RNA that targets this repetitive sequence that's repeated throughout this entire region. And so this is an example of what this looks like in cells. So here's an example where the SUNTAG marks the Y chromosome in the interphase nucleus. And when we induce centromere inactivation and watch cells go into mitosis, you can see that these Y chromosomes rapidly missegregate or misaligned. So here's an example of a misaligned Y chromosome from the metaphase plate. And when these cells segregate their chromosomes in anaphase, this misaligned Y chromosome undergoes sister chromatid disjunction. One of these sister chromatids ended up back in the nucleus, and another one ended up in this micronucleus. And so now we have a very nice way where we can tag the missegregated chromosome and watch it missegregate into a micronucleus. And so the question that we really wanted to answer is what happens to this chromosome during the next mitosis, where it would undergo fragmentation, for example, by the activation of the Fanconi anemia pathway. So here's an example of a Y chromosome inside of a micronucleus. This cell would now go into mitosis, and what we had expected, or what I had initially expected, was that the fluorescent signal from this sun tag would essentially disperse all over the mitotic cytoplasm. But that's actually the complete opposite of what we saw. So in every instance of a cell entering mitosis, the fragmented chromosome in the micronucleus remained clustered throughout the entire duration of mitosis. Um, and this was actually quite unexpected, at least for me. And so these chromosomes, as you can see, they cluster in the single location in the mitotic cell. And when these cells segregate their genomes during anaphase, importantly, the vast majority of these fragments are now inherited by a single daughter cell. So there's a mechanism that seems to intrinsically cluster these chromosome fragments together, and this can bias the way that they are inherited during mitosis. And here you can see the fragmented pieces are now reincorporated back into one of the daughter nuclei as what is referred to as a micronucleus body. And so to confirm this in fixed cells, we also did experiments where we can take cells at different stages of mitosis, stain the Y chromosome for gamma H2AX as a marker to look to see which ones are fragmented Y chromosomes. And as you can see, most of these control Y chromosomes are all intact and clustered together as you would expect for a regular chromosome. But if you now look at gamma H2AX positive Y chromosomes, and these are the ones that are likely to be fragmented by a Chromothripsis event, you can see that again they are also indeed clustered together. And if you look at cells after anaphase or in telophase, you can see that these fragments are indeed inherited by only a single cell rather than by two cells. And we can also design methods in which we can quantify this based off of the intensity of the Fish signals that are inherited between the two daughter cells. And we can show that these cells, instead of undergoing a 1-to-1 equal segregation ratio, they typically undergo a 0-to-1 unequal segregation ratio, or approaching 0-to-1. Okay, so if most of these fragments are inherited by one daughter cell, this would lead to the prediction that in examples of Chromothripsis in cancer genomes, there would be examples of Chromothripsis events in which there are minimal DNA copy number changes. And so this is an example of a canonical Chromothripsis event in which there are a number of rearrangements, and there's also this oscillating DNA copy number step that represents the loss of these fragments. However, we then teamed up with Isidro Cortes Soriano at EMBL-EBI to look for events in which there are Chromothripsis, but there's a lack of DNA copy number losses. And so here are a couple of these examples that we found. And so, for example, this is a example of a tumor with Chromothripsis of chromosome 1, in which there's a series of complex rearrangements on this chromosome, but the copy number state is at a completely neutral copy number state of 2. And so if we look at these events in pan-cancer genomes, what we can find is that these balanced Chromothripsis events that maintains their copy number occurs in approximately 5% of all cancer genomes. And this is also, we also found numerous examples in which these events can disrupt known cancer drivers or also generate recurrent oncogenic fusion genes. So the next question is then, how exactly does this happen? So there's a couple of potential candidates that can potentially tether DNA breaks during mitosis, and this includes DNA polymerase theta as part of the alternative end-joining pathway, the MRN complex, which is comprised of these three components, as well as the CYP2A top EP1 complex that can tether DNA breaks during cell division. And so to determine whether any of these factors are responsible for tethering these fragmented chromosomes in mitosis, we deleted each of these components in cells. And what we saw was that in cells that lack pol theta or NBS1, the chromosomes remain clustered as you would expect from similarly to wild type cells. But if you now look at cells that are deficient in CYP2A or its interacting partner top EP1, we can see that there's a very strong increase in the dispersion frequency of these chromosomes. So instead of remaining clustered together in an intact region during mitosis, these chromosomes now disperse all over the mitotic cytoplasm. Here's another example of this. So this is a clone that is a CYP2A deficient cell from a CYP2A deficient cell, and after we induce Y chromosome missegregation and chromothripsis, we can see that there are fragments of the Y chromosome that are now pushed away from the primary genomic mass, and all of these are also marked by gamma H2AX, suggesting that the CYP2A top EP1 complex can tether these fragments together during mitosis. Once the cells come out of mitosis, for example, in the absence of CYP2A, in which they fail to properly tether the fragmented pieces together, these fragments can then accumulate within the cytoplasm of the daughter cells. So, for example, this is a cell in which we've semi-permeabilized the cells, and so we're now just looking at the cytoplasm, and we stain them with an anti-double-stranded DNA antibody, and you can see the appearance of these double-stranded DNAs within the cytoplasm. Additionally, we can also confirm this by FISH that it's indeed specific for the Y chromosome. So if we're using a Y chromosome probe, we could see these cytoplasmic Y chromosome fragments. But this is not true for for a control chromosome, for example, using the X. And we can exacerbate these conditions in cells that are deficient for CYP2A, but we can also see this particularly as well as in wild-type cells. And this, additionally, we can also, these fragmented pieces that now reside in the cytoplasm harbor DNA damage marks. So for example, if you stain with gamma H2AX, they still are chromatinized. So they contain these DNA damage marks that were carried over from the previous cell cycle. And we can also show using staining for lamin B1 that some of these fragmented pieces in the cytoplasm actually lack a nuclear membrane. So from these studies, we had proposed this sort of model in which there's a very strong DNA replication defect during interphase within micronuclei that then activates the Fanconi pathway during mitosis, and this induces fragmentation of the chromosome. But these fragments are held together by the CYP2A top EP1 complex. This biases the inheritance of these fragmented chromosomes such that only one daughter cell receives the vast majority of these fragments, and this minimizes the loss of DNA copy numbers during cancer genome evolution to give rise to the signature that we term balanced chromothripsis. These chromosomes within now that resides in the nucleus of these daughter cells are referred to as micronuclei bodies, also begin to repair these chromosomes using the cell's DNA repair machinery, particularly the non-homologous end-joining pathway. So here in this experiment done by Ching in my lab, so if we take cells and mark the chromosome in the micronucleus using the Suntag system, and then track it over time in the presence of a DNA repair reporter, for example, a halo tag version of 53BP1, we can see that after mitosis, 53BP1 begins to accumulate within approximately 10 hours of interphase on these fragmented chromosomes. And as they become repaired during that subsequent interphase, this 53BP1 signal now declines. In the absence of CYP2A top EP1, these fragmented pieces now disperse all over the mitotic cytoplasm, and this can also then result in the formation of these cytoplasmic DNAs that can then activate potentially the cGAS sting pathway, for example. And then in the final couple of 15 minutes or so, we next wanted to ask what happens to these chromosome fragments in the cytoplasm. This is work from a very talented PhD student in my lab, Liz. And so Liz set out to answer the question of beyond activation of cell intrinsic signaling programs, such as the cGAS sting pathway, can cytoplasmic DNAs also elicit non-cell autonomous responses to nearby cells? And so we now have significant evidence to suggest that these fragmented chromosomes from micronuclei can in fact accumulate in the cytoplasm. And so to answer this question, she did a series of relatively simple experiments in which she took RPE1 cells, so these are diploid human cells. She stained the DNA with cDNA, which is a fluorescent DNA dye, and then she treated cells with a combination of a SEMP inhibitor and an MPS1 inhibitor to induce mitotic errors. And so this is a time-lapse movie, and I'll point your attention to this cell here with this micronucleus. And as I let the movie play, you'll see that the cells move around as cells normally do, and they can also form these stretch-long protrusions between two different cells. And if we look at these protrusions at a particular snapshot, we can see that these cells are indeed connected by this long tube-like structure, and that if we then look at the DNA signal, we can see the presence of this DNA that appears to transfer between these tube-like structures. And so from looking in the literature, these are referred to as tunneling nanotubes and have been well documented in a number of different physiological and cancer-associated contexts. So for example, these nanotubes can be formed between two different cells for example, in the tumor microenvironment, and this can mediate the transfer of different cytoplasmic cargo. And what has been demonstrated so far is that different proteins, organelles such as mitochondria, and also RNAs such as mRNA and microRNAs can transfer through these tubes and mediate different cellular interactions. And so one outstanding question is whether DNA, if it's present in the cytoplasm, whether this can also serve as cargo that can be transferred by these nanotube structures. So cells move around in culture quite a bit, and so this really makes it challenging to really capture these tubes because they're so dynamic. And so one trick that we then started to do is treat cells with a low dose of acetylcholine D, which is an actin inhibitor, which is an inhibitor of actin polymerization. And I'll let this movie play, and what you can see is that the cells immediately stop moving, but importantly, the cells that have formed pre-existing tubes continue to maintain those tubes. And if we now look at these cells containing these nanotubes, you can see the passage of cargo that's being transferred from one cell to another cell. And so using this approach, if we now combine this with staining of DNA after the induction of mitotic errors, we can find examples in which these cells that form this tube begins to transfer this DNA from one cell to another. And if we now take these cells and stain them with different markers of the cytoskeleton, for example, alpha tubulin and beta actin, we can see examples where there are tubes, these nanotubes that form between adjacent cells, and they can also entrap DNA if we now stain these cells with yo-yo-1. And this is very different from chromosome bridges that arises as a defect from mitosis. So for example, if we take cells and we induce the formation of dicentric chromosomes, and which results in the formation of interphase chromosome bridges after defective mitosis, we can see that there are that these interphase chromosome bridges are also marked by the components of the cytoskeleton, including tubulin and actin, but the morphology of the underlying DNA of the stretch bridge is very different. So this is a very stretched apart bridge connecting two daughter cells. And then this example is a piece of DNA from the cytoplasm that connects two unrelated cells. And so this led us to this very simplistic drawing where cells that a genomically unstable cell can form a nanotube with another cell and through these nanotubes, these DNAs can now passage through these nanotubes to transfer from a donor cell into a recipient cell. And if you now take these, and what we wanted to do is establish that these are in fact unrelated cells. So what Liz did was she took cells and she marked the chromatin in one of these cells using H2B GFP, and then she marked the chromatin of the recipient cell using a different fluorescent color, also expressing histone H2B, in this case, M cherry. So this is an example in which we've now cultured these two independently labeled cells, so RPE cells with H2B M cherry and RPE1 cells with H2B GFP. And we can see that when we let this movie play, you're going to see a green cell with the green micronucleus coming up from the bottom of the screen. And that green micronucleus will now seemingly transfer over to a cell that contains a red labeled nucleus. So this is, and we've now seen multiple examples of these types of events in which the DNA, or at least the chromatin here, can transfer from different distinctly labeled cell populations. And we can also do these experiments in different cell types. So this is an example of a renal proximal tubule epithelial cell from the kidney with retinal pigment epithelial cells. And we can also identify a number of these different events in which these mismatched colored micronuclei appear to transfer from one cell to another cell. So under these conditions, we would expect that if we now take these cells and stain them for markers of the cytoskeleton, we can also see these events becoming incorporated into the cytoplasm of the recipient cell. And so this is an example where we took these RPTECs, these kidney epithelial cells labeled with different H2B colors, and we can see that this is a RPTEC cell with a red nucleus, and now we can identify that it also can contain fragments or that are differentially labeled that presumably transfer from a green labeled cell. And this is following the depletion of MAD2, which is a component of the mitotic spindle checkpoint, in order to induce mitotic errors in these donor cells. Additionally, we can also do these experiments by arresting cells in mitosis using nicotazole and then releasing them, and they can then form inappropriate kinetochore microtubule attachments to also generate mitotic errors. And we can also, in fact, see these examples of mismatched labeled DNA within these recipient cells. So presumably, these green fragmented pieces came from a different cell and transferred over to this cell with a red nucleus. Here's another example of this. So in this example, we're mixing two different cells depleted of depleted of MAD2. And for those of you who study micronuclei, you can look at these images of DAPI stained cells, and you would almost immediately think that this micronucleus belongs to this cell with this nucleus. And if we now look at the different channels of H2B, we can see that this nucleus is actually a red nucleus, and this micronucleus is a green micronucleus, suggesting that this micronucleus had in fact transferred over from a green cell. We can also identify many examples in which the transferred material contains both fluorescent histone H2B markers, and this presumably arises from DNA replication in the recipient cell and drives the replacement of different histones during replication. And this is quantified here. So looking at over 1,000 cells with micronuclei, we can see the vast majority of these cells had undergone replication after the DNA transfer event and produced this mixed H2B signal. Additionally, we wanted to next look at different ways that this can be induced. So here's an example where we took these RPTEC cells and we induced micronuclei by triggering the formation of DNA double-strand breaks using Cas9 mediated genome editing. So here we're inducing a DNA break on human chromosome 3, and this would then produce a fragment of chromosome 3p that would then mislocalize to this micronucleus. And if we do these co-culture experiments, we can in fact see many examples of these chromosomes that are mismatch labeled transferring between different cells. Additionally, we can also take cells and hit them with ionizing radiation to induce genome-wide DNA double-strand breaks, and we can also indeed find examples in which there are these mismatch labels between different cells. And then so in another experiment, Liz next wanted to determine whether that DNA that had been transferred truly resides in the cytoplasm of the recipient cell. So this is a little complicated experiment, but I'll try to walk you through it. So in this system, she's taking female cells. So these female cells do not have the Y chromosome, and she's introducing the dCas9 SUNTAG reporter that I had described earlier. And so using this system, we can target dCas9 SUNTAG, which recruits GFP, to a specific sequence of the Y chromosome. So in female cells, this SUNTAG reporter usually does not light up. So now she's taking male donor cells containing Y chromosome micronuclei using a variety of different methods. And the chromatin in this donor cell is labeled with H2B mCherry. So the expectation is that if you now co-culture these two cells together, this red micronucleus would then, from a male cell containing the Y chromosome, would transfer over to a female cell. And if this was in fact the Y chromosome, then this would illuminate or light up the cytoplasmic Y chromosome reporter in the recipient cytoplasm. And so this is an example of this in action. So here's an example where you have this cell with that's been stained with DAPI, and we can also look at the plasma membrane using this marker called Z01. And here's an example where we can see this DAPI stain material is also positive for H2B mCherry, so it presumably arose from the male donor cell. And this, in fact, lights up or activates this cytoplasmic dCas9 SUNTAG reporter that's specific for the Y chromosome in the female cell. So just to reiterate, this is a female cell in which the Y chromosome had now entered the cytoplasm and then activated this internal reporter. Next, we wanted to ask whether the transferred DNA can potentially intermix with the host genome. And so if the cells go into the next mitosis, we would imagine that all the chromosomes would condense and then potentially mix together. And so next, Liz looked at metaphase spreads, and she looked for examples of these mismatched DNAs. And indeed, she can identify numerous examples in which there are presumably what looks to be an entire intact chromosome, as well as fragmented pieces of a chromosome that can transfer between these two cells that produces this mismatched H2B GFP labeling. And in a complementary experiment, she wanted to then directly label the DNA itself. So she used nucleotide analogs in populations of cells labeled with IDU and another population labeled with CLDU, and then she co-cultured these and then looked at metaphase spreads. And so indeed, we can also, Liz can also see numerous examples in which these IDU containing fragments, for example, can be found in a cell that contains a CLDU labeled genome. Okay, so one final series of experiments. So we next wanted to ask, are these transferred DNA fragments stable and functional in the recipient cell genome? And so in this experiment, Liz, these events are quite rare from our measurements. And so she wanted to find a way where we can induce this on a specific chromosome and then isolate or enrich for these events afterwards. And so to do this, we're going back to our Y chromosome system that I described earlier that importantly has its neomycin resistance locus on the chromosome. And so we've demonstrated previously that this system can in fact produce Y chromosome micronuclei as well as cytoplasmic DNA fragments that contain the Y chromosome. So using this strategy, Liz devised this experimental schematic in which we can take these cells that I just described and we labeled these cells with green fluorescence. And then we can mix these cells with female recipient cells. And these cells are also labeled with different fluorescent markers. So they're labeled in red. They have a different set of resistance markers, zeosin, and these are HeLa female cells. And so she's co-culturing these two different cells together. She's adding doxycycline and auxin to induce fragmentation of the Y chromosome in the male donor cell. And the expectation is that these fragmented pieces would then transfer over to these female recipient cells. And if true, these cells would now become resistant to that neomycin resistance selection. She then subjected these populations of cells to co-selection with these two different markers, and in the end, we're left with three different populations of cells. I'll quickly go over these, but one of these populations are cell-cell fusions, which causes both red and green cells, or essentially a yellow cell. And also, additionally, the population of interest are these M-cherry positive cells. So these are the female cells that have now become resistant to the Y chromosome. Or became resistant to the selection marker encoded on the Y chromosome. So after Liz induced these conditions and then looked at them by flow cytometry, she can see that in untreated cells, so these are control cells, most of these events generate cell-cell fusion events. So these yellow cells simply due to co-selection of the two antibiotics. But if we now treat the cells with doxycycline and auxin, she can shift this spectrum of cell-cell fusion events to now become more M-cherry positive cells in these co-cultures. So what this suggests is that after induction of Y chromosome fragmentation in the male cell, you can now enrich for these female cells that had now incorporated the Y chromosome fragments. And this is quantified here. Next, if she looks at metaphase spreads, she can see that under control conditions, they are indeed comprised mostly of cell-cell fusion events. So if you count up the total number of chromosomes in each of these cells, they essentially represent a doubling of the genome from the male donor cell and the female recipient cell. And if we now look at the M-cherry fraction of cells that we are interested in, these cells maintain a karyotype that's almost identical to the female recipient cell, suggesting that they did not undergo cell-cell fusion to become resistant. We can also use PCR to detect fragments of the Y chromosome in these female recipient cells as well. And then in one final experiment, Liz then took these cells and then expanded them up and then performed cytogenetics by metaphase FISH. And so in these experiments, she collected these metaphase spreads from these female recipient cells and then used either a Y chromosome paint probe to mark the Y chromosome or using a back probe that marks specifically the neomycin resistance locus on the Y chromosome. And so here are examples of female metaphase spreads that contains the expected number of chromosomes from the parental female line, and they contain fragments, and strikingly, they contain fragments of these Y chromosome sequences in their genomes, suggesting that these transferred DNA fragments are functional because they can encode genes that reside on them. For example, the neomycin resistance gene, and they are also stably maintained for a long period of time as these extra chromosomal DNAs. And so if this were true, then I think these findings would advance our understanding of cell-cell interactions, including the potential transfer of oncogenes and or deleterious mutations from chromosomally unstable cancer cells to non-cancer cells in a shared tumor microenvironment. And so just with that, I'll conclude with a couple of outstanding questions. So some areas that we're now focused on is understanding how after nuclear envelope rupture, how do these fundamental chromosomal processes such as DNA replication and transcription occur in an environment with limited nuclear factors? What are the replication intermediates that engage the FA pathway or the CYP2A top BP1 complex in mitosis? Could targeting the FA pathway represent a strategy to prevent the emergence of ecDNA-mediated resistance to cancer therapies? How did distinct patterns of chromothripsis emerge during the genomic evolution of different cancer types? And currently we're focused on clear cell renal cell carcinoma, and hopefully I can tell you about that next time. And also, lastly, intercellular DNA transfer. Very, very odd, but how does it happen and why does it happen? What is the significance of this? Okay, so with that, I would just like to thank the members of my lab. So today I showed a lot of work from Justin, Yufeng, and Liz, but really the entire lab really contributed to the majority of these projects, as well as the number of outstanding collaborators. And I'll highlight Isidro, who's had his hands on pretty much every project in our lab. Roger Lowe for his collaboration with the targeted therapy experiments, as well as our sources of funding. And so with that, I'm happy to take any questions.
Peter, thank you very much. That was excellent. If you have questions, you can just simply type them in the chat and I read them out, or you unmute and ask them yourself if you're very, very brave. To keep us busy until then, there's Jeff. Jeff is in a second. Oh, Jeff, do you want to go? Okay. I just have to say, I really hate the last part of your talk, Peter. I just so hate it. And the reason I hate this part so much is because we build little phylogenies of single cells. We build these little trees. And the one thing we explicitly exclude is that these cells kind of exchange stuff between each other. So all the changes we put into the ancestral, in the recent ancestors. And so you're crashing all our computational models. Unless what you're describing is very, very rare and it doesn't actually matter. Is it?
So we think it is a very rare event, just based on our ability to measure it. But we think that under certain conditions, this could potentially impact the way cancer genomes evolve.
Yeah.
And so hopefully people like you can devise algorithms to potentially detect this and determine how frequently it actually occurs physiologically.
Okay, good. Thank you. Jeff.
Beautiful talk, Peter. I'm going to pick up on the DNA transfer as well. Why would this occur from an evolutionary perspective? Why are the cells maintaining this ability to transfer DNA, do you think? Is it accidental, or is there a need to maintain this function?
Yeah, that's a fantastic question, and great to see you, Jeff. I think the answer, so we don't know for sure, but I think the answer is that it's accidental. So various components in the cytoplasm can in fact transfer between these tubes. And we think that under conditions of genomic instability, in which you can have DNA in the cytoplasm, which normally doesn't occur under normal circumstances, then they can go along for the ride in the transfer event. And so now we're doing experiments to determine whether this is a directed active mechanism of transfer or it's just a passive mechanism. But usually DNA is in the nucleus, and so it's protected from being transferred. But in different conditions, particularly cells with DNA damage, where after they experience some type of mitotic defect, you could then have DNA in the cytoplasm that could then serve as cargo for transfer. Yeah.
Right. Excellent. Serena writes, really fantastic presentation, Peter. So great to hear your recent intriguing work. So thumbs up from Serena. Anybody else? Any questions? We would have time for one more. If not, no problem either, because Peter, thank you very much for keeping on time. We are very good on time right now. So thank you everyone for coming to this lecture series. And Peter, thank you very much for an excellent talk that will keep us all thinking for a while.
No problem. Thank you very much for having me.
And tell your friends if they couldn't make it to the talk, I'll put it up on YouTube soon. Thank you very much. Bye,
Thank
Peter.
you.
Bye, all of you.